Okay. So good evening, everyone. My name is Seemanti Aditya. So I am the NPTEL TA for the course Cell Culture Technology, and I usually take the sessions from 6 p.m. to 8 p.m. every Monday. So today we'll be discussing about uh, what uh, what the last week's lectures have been about, and towards the end we will be solving some assignment questions, also some not this year's assignment questions, like some practice questions, and also we'll be discussing about any doubts or any other queries that you have, so at the end of the session. So let's begin. So, uh, so today in this week's session, we'll be discussing about what are the different basic cell culture requirements, what equipments do we have in cell cultures lab, and what are the necessary precautions that we need to take. What are the different kind of issues that are normally, which are generally present in case of in generally all the cell culture labs around the world. So let's begin. So what are the requirements, what are the cell culture facilities requirements that we have? So and based on those requirements, the newly joined professors, they will design the lab. They will design the cell culture lab based on what kind of techniques or what kind of expertise they are working on. So uh, first and foremost point that we need to keep in mind is what level of facility do we really require. Do we require a very high level of facility where we would be working with viruses, or where we would be working with some dangerous bacteria, or whether or do we require some basic cell culture lab where we are not concerned with the dangerous microbes. So based on that, there are different levels of facilities. There are like advanced level of cell culture, there is like basic level of cell culture, there is the medium level of cell culture, and so depending upon what are the requirements and what amount of what are the sources of funding that you have. So different people, they wish to design the lab in such a way so which will meet all their requirements and also will suffice the funds, and will be like inside the amount fund that is been assigned to develop the cell culture lab. So depending upon those facilities, whether they are working on the cell biology or developmental biology aspect. So cell biology and developmental biology aspects basically means that different kind of development of like organisms, whether we want to study the development of any kind of organism from the embryo stage to the adult stage. This is generally people can use for insects like Drosophila, or maybe fishes like zebra fishes, or so. These model organisms, these are very, very easy to study for development purposes, because they are very easy to grow, very easy to handle, and economically also they are very cheap. So those kind of facilities, if they require to culture those insect cells, or they require to culture the embryo of some fishes, then those facilities are required. Also cell biology, those people work with extensive cell biology signaling processes, or with extensive microscopic imaging, or with different kind of cell mediated assays they require. So those kind of labs will require cell biology techniques, or cell biology lab which is not very, which is not, it's not, it does not have to be really very sterilized or really very advanced level. But yeah, just to study the mechanisms, medium level of cell culture facility is fine. Also, people work with cell-based biosensors. So, what are cell-based biosensors here? The cells which are genetically engineered, and they have certain genes inside the cells which can express like some kind of fluorescent protein, which some kind of fluorescence protein, or some kind of sensing mechanism through which a lot of drugs or a lot of analytes can be analyzed using this kind of sensors. So what the cell-based biosensors mean is, these cells are not generally the cultured cells which people generally use, not the normal kind of cells which is directly isolated from the tissues and grown, but these are some kind of genetically modified cells. So these are genetically modified cells. Okay. So genetically modified cells where they have certain kind of genes already expressing inside their genome. So when we are using certain kinds of biosensors like this, we require some little bit advanced level of cell culture, because we do not want any kind of contamination, or we do not want any kind of issues which will destroy the cells or which will kill the cells, because we cannot afford to regularly make genetically engineered or genetically modified cells. These are very difficult to make, because genetically, any kind of genetically altered organism, it is very difficult to grow. They require a lot of attention, they require a lot of changes in the composition of media, they require like different kind of specialized needs, which is not possible on a day-to-day basis if an entire set of cells get killed because of contamination issues. Contamination is a very, very big problem in cell cultures, because practically anything and everything that we are surrounded with is has microbes in it, and we need an extremely clean room for performing cell culture, because those microbes then if somehow they get entry into the cell culture room, then it's going to create nuisance, and the cells growth will be affected. Also, not only just the growth, but also the properties of the cells are affected. Next kind of facility that generally people require is cell tissue engineering. So this is a new field, and this is now a very hot topic, because what people do is they collect some cells, and they perform regenerative medicine or tissue engineering, in the sense that where people claim that they can make the heart or kidneys or the lungs in the lab. So basically, creating a tissue from a mass of cells, or different masses of cells. This also requires a very, very high level of cell culture, because cell culture labs, because we require a lot of sterilization techniques. If you are making a heart inside the lab, and we want that heart to be replaced in a in vivo system, then that needs to be really sterilized. It should not have even one bacteria or even one microbe in it. So the cell tissue engineering labs, or the regenerative medicine labs, they will require a very advanced level of cell culture, with very high level of sterilization techniques, and very sophisticated cell culture incubators and hoods, which is specialized for like according to their needs, and are not easily affected by any contamination. So next kind of people, what they do with cells are the labs that work on screening of different drugs. So basically, these labs they are not very much interested in generating a particular tissue or an organ from cells, or working on some kind of sensors or those things. They just want to test whether the drug is working on the cells, whether the drug is showing positive effect or negative effect or no effect at all. So this kind of labs, they do studies on a lot of drugs. These are like maybe at least hundreds and hundreds of drugs. So these are in larger scale. So we do not. So here it is not required that we need a very very sterilized room. But yes, medium level of sterilization is required to test drugs that are such larger quantity. So this kind of facilities are required. Apart from that, another kind of people who work with cell culture systems, development of cell culture systems. So these are people who work on the cells basically for their different experiments, designing some drugs, testing the drugs, discovering the signaling mechanisms, or generating a whole tissue or an organ. But apart from that, there are different kind of people who work on how to improve the cell culture systems. So how to improve the cell culture systems in the sense that how to make consumable items which are much more versatile with the cells that we are using. Like they have all kind of media development like programs where the media that we use for cells, generally what happens is there is DMEM media, the Dalbecker's modified eagle medium. DMEM is the medium that we use generally. It's known as DMEM. So generally in maximum kind of cells, so this medium contains all the possible basic amino acids and the salts and nutrients and glucose and whatever is required for the cells to thrive. So this medium contains maximum of all the things, except for few differentiating agents or few growth factors that is required in some specialized cells, some specialized neuronal cells. All other factors are present in this kind of media. So people who work on media development actually work on how to improve the quality as well as the product economical value by causing less toxicity to the cells and mimicking the physiological environment. So it is very important that this media mimics the physiological environment in which cells actually are present inside the cell, inside the body. So it should mimic the physiological environment. Physiological environment.
Ma'am, what are cell-based bio-MEMS?
The cell-based bio-MEMs are biosensors, and what is the full form is micro environment. What is it called? Micro electromechanical systems. So bio-MEMs basically is used in case of like lab on chip techniques, where different kinds of cell based like basically these are actually kind of chips which has like has activities of kind of biological cells, where this will mimic a environment inside the cell, but is actually a mechanical part, and like has a mechanical part and also has a biological part. And it can it is mainly used for testing of drugs. It is mainly used for large scale analysis of different kinds of compounds. So these are not like cell cultures, but it is generally a chip which has like kind of small chip which has some cells you grow and test whether it is suitable for different drugs or not. So it's like a mechanical, it's like a conjugation of mechanical engineering and biology. So these are bio-MEMs. So yeah. So yeah, so people who work with this kind of media development is like they can mimic the physiological conditions, like the media should mimic the physiological conditions inside the body, and also they should have at minimum cost they should be able to provide a lot of benefits in terms of cell growth and in terms of cell proliferation. So, development of cell culture system also includes increase growth of substrate and cell-based bioMEMs that we talked. Like cell-based bioMEMs basically is like a lab-on-chip method where they have some chip where they can grow some cells and then test some kind of drugs or analytes like that. And also substrate means what kind of substrate we can provide to the cells so that they can benefit from those substrate and grow, and they can collect nutrients, so those kind of things. Then we need to have also a regular maintenance of cell lines. Like apart from all these facilities, the very important thing is to maintain the cell lines. So whenever we are isolating cells from tissues, it is very important that this cell line that we have created needs to be maintained properly, because they are they depend, depending upon what is their growth rate, they can grow very fast or they can grow very slow. And so depending upon what are the requirements, we need to regularly change their media, because their media gets depleted, and we can see that the media is generally red colored, and if the media color turns yellow, that means the media is now depleted of the nutrients, and it is now like the cells also secrete a lot of toxic chemicals. So those toxic chemicals, their excretion is now present in that media. Now those media needs to be removed. Also, if the cells are overgrowing and then like it is totally full, it's totally covering the surface area of the dishes or the flasks that we are growing it in, so we need to split the cells. That is also known as passaging. And if we want to freeze it, then that is called freezing. We need to freeze this if we want to if the cells need to be like frozen and kept, and we want to use it for later purposes, then we have to freeze it. So these are some of the requirements and some of the like general practices of cell culture. And so apart from that, we have to so apart from that, we have to see how the cell culture room typically looks like. So the cell culture room so the cell culture room looks like this kind of. So this is like it has a hood, you can see this is the hood, and this is the cell culture incubator. So this is the incubator in the sense that it has carbon dioxide in it, and it helps in maintaining the buffer carbonate. So inside our body we have the biocarbonate buffer system. So in this kind of cell culture incubators, carbon dioxide is supplied through this this cylinder. So these are carbon dioxide cylinders, and here you can see on the top of this carbon dioxide cylinders, you can see how much pressure is being maintained. So it is also very important that when we are attaching the carbon dioxide cylinders to the incubators, this pressure has to be at one particular level, so that it is not very quickly refilling the incubator. So these are very automated systems. Whenever the carbon dioxide level drops, this pressure valve gets so this pressure valve can detect the changes in the carbon dioxide level and will refill it. But we do not want it to get refilled very fast, or we do not want it to get the carbon dioxide should be depleted very fast. So those pressure settings and all is already standardized and optimized for all of the cell culture, where the 5% carbon dioxide level is maintained in this carbon dioxide incubator, along with the temperature at 37 degrees Celsius, which is the normal physiological conditions inside the cells. So these carbon dioxide incubators, depending upon how many people are actually working and how many cell lines are being maintained in a particular lab, people can have a lot of incubators. Here you can see 2 cell culture incubators and also a cell culture hood. So this cell culture hood is basically the vertical hood where the airflow is from the top. So the HEPA filter, so there is a term called as the HEPA filter, which is like used for, filtering smaller particles, and small contaminating prone microbes. So those HEPA filters are placed in this particular region. So basically in vertical airflow system, here the HEPA filter is placed, and the air is passed from here. So all the air that comes out is totally sterile, and it is not having any kind of contamination. Ideally, it should not have any kind of contamination. But this HEPA filter needs to be changed very regularly, because this HEPA filter gets like filled with all these particles and contaminating microbes. So HEPA filters need to be changed ideally after every 6 months or 1 year. So depending upon how if sometimes some cell culture hoods have HEPA filters which can clean itself, self cleaning filters, but those are very expensive. But generally what people use are like the HEPA filters which are replaceable, like once it is contaminated or once it is it has been used for quite some time, then people can replace it. So cell culture hood is very important for performing all the cell culture activities. So we need to perform everything inside this system. Like whenever you can see this glass door, so this glass door has to be opened, and it has to be not open to a very big, like very high level. It should be just open to this level, so that just you can insert your hands through this portion and perform your work. So it is a skill that people can gain over a period of time when they're doing cell culture, that we cannot lift it up too much, but we have to do everything within this this space, because if we are lifting it too much, then we can like the air from outside can have a like access to enter the system, although there is HEPA filter and all, and the airflow is in vertical direction, but still there is chances. So people ideally prefer to not open the glass too much and just insert their hands and do all their work and then again close it. Apart from this hood and the incubators and the carbon dioxide cylinders, also very important thing is the microscope. So this microscope, one microscope is very important inside the cell culture room to actually observe how much the cells have grown in the system. So to observe the growth of the cells and to see whether you have to give a media change to the cells, or whether you have to passage the cells, or whether you have to freeze the cells. So this microscope inside the cell culture room is very important. All the cell culture flasks and the tissue culture dishes have to be kept inside this incubator. So and whenever we are taking it out, we have to be very careful to always wash our hands and always use the 70% ethanol spray, wipe our hands properly, and then open the incubator, take out the cells. And also whenever we are checking the cells in the microscope, we generally prefer to clean the surface near to the microscope with 70% ethanol. Even on the space where we are keeping the cells, we have to clean it with 70% ethanol, and then place the cells on it and check what we have to do with the cells next. So apart from this, we have all kinds of other things: centrifuge, vortexes, cells counter. So one thing is cell counter. What cell counter does is you have to take some amount of media or some amount of cells in a cube, and then just put it inside the cell counter, and can count how much amount of cells are there in your sample per ml or per microliter. So depending upon that, like cell counter is generally required for when we want to transfer, when we want to have equal number of cells for all the conditions. Suppose we want control and treatment and different treatment 1, treatment 2, every other condition, we need to have equal number of cells. So for that, we need to seed the cells in such a way that it has to be equal in all the 3 flasks. So there we have to see how much cells are there, and then we have to actually use our cell counting technique to see, okay, in one field we have 10 cells, so in total area of like if the area is 35 mm, so in 35 mm how much amount of cells I will have if I am seeding it in that way. But what we generally prefer to do is we don't count the cells. We pellet down the cell, and then we pellet down the cells in such a way that when in the pellet we will add suppose 300 microliter of media, and in 3 flasks, if we want to split it into equal amount, then out of 300 microliter, 100, 100, 100 we will split into 3 dishes. So what will happen is all the 3 dishes will have equal amount of cells, because so if we are passaging the cells from 160 mm to 360 mm, and we want that 360 mm should have equal number of cells. So 160 mm, if we are pelleting the cells, and in that cell pellet we will add 300 microliter of the media. These are spending the cells in 300 microliter of media, and then put 100, 100, 100 microliter each in each of the dishes. So we will have equal amount of distribution of cells in the 3 different flasks. So then, apart from this, the cell culture room have to be very, very clean. As you can see that there should, the floor, the benches, everywhere, it should be very clean and very much sterilized. So outside the cell culture room, people generally prefer to keep shoe racks and the lab coats and headbands and the gloves, etc. So we have to wash the hands, keep our shoes outside, wear the lab coat, wear the head cap, and wear, sometimes also people prefer to wear mask, because even if you're talking, the droplets that in your so if you're talking and the droplets are surrounded, so whenever the droplets are getting surrounded, so those droplets also contain microbes, and those can also create issues. So we never know, so it's always better to take precautions, and always better to carry out cell culture in a very, very in a very sterilized environment, and we have to be really very careful. So apart from that, what are the cell culture requirements that we have? So cell culture requirements requires a lot of space, because you can see that there is so many instruments need to be accommodated in a space, and we need a lot of space for that, and we do not want any kind of misfortunate events to take place, because this incubators and the cell the carbon dioxide cylinders and all, these are really heavy items, and they require space to be stored, and they cannot be moved from one place to another. So once they are settled in one particular portion, we do not want it to move again and again. And so this needs to be really spacious, and also the management have to be very proper, the space management. So it has to be used very properly in such a way that no instrument is blocking the exit or entry pathway. No instrument is, having like too much of free space in between. So it has to be placed like incubators, hood, and then the carbon dioxide cylinders and all the liquid nitrogen vessels. Everything has to be in a particular order, so that there is no space blockage, or there is no misuse of the space. Apart from that, all the funds and finances that is required for setting up a cell culture lab is also very huge, because each of these instruments are around lakhs of rupees, and they need to be maintained with a lot of care, because a little bit of carelessness can lead to loss of lot of money and a lot of time, actually, because this skill and this technique is not very time convenient. It takes a lot of time to culture the cells, to maintain the cells. So just a small mistake or a small carelessness can actually cost a hefty amount of not only money but also time and your patience. So finances and funds are a major issue in like all of the places, but yeah, in experimental labs, basically where we require this much of sophisticated instruments and so much of consumable items, it is very we have to judiciously use the finances and funds that is provided. Also, what kind of equipments people require, whether they require vortex, whether they require water bath, whether they require some kind of centrifuge, like higher centrifuge higher speed centrifuges. Whatever centrifuge we use inside the cell culture lab is slow speed centrifuge, just to pellet the cells. So, but sometimes people require ultra centrifuge for subcellular fractionization and all. So what kind of requirements people have, depending upon that, people can use different kind of equipments, and there are higher levels of sophisticated equipments that are present for cell culture. There are room HEPA filters, there is room UV, there is room totally sterilization, one air flow is there. So whatever system people require, depending upon that and depending upon how much finances they have, and they can invest on cell culture labs, they can, actually build the cell culture facility that way. Also, apart from that, the plan for expansion and modification in the sense that, so from 10 years, so if we are building a cell culture facility now, 10 years from now, there will be much more sophisticated or much more advanced instruments that will like make the life easier. So always there needs to be some kind of free space or buffer space that will accommodate that uh, like advancement, like that will accommodate the advancement of technology, so that we can incorporate that in the cell culture lab. So always it is better to have a plan for the future, not just for the present, because always the science will advance, and we always need to be prepared for what is coming next. Also, depending upon the level of biosafety, BSL 1, BSL 2, 3, and 4 levels of biosafety is present, where each of these levels, like BSL 1 is the lowest and BSL 4 is the highest and advanced level of biosafety. BSL 4 level of biosafety is generally required for very infectious agents like viruses, for SARS-CoV-2 or Ebola viruses. BSL 4 facilities or BSL 3 facilities are required. BSL 3 is required for mycobacterium bacteria. So this kind of facilities are not available everywhere, and also special permission is required if we have this kind of higher advanced level of facilities. So in generally, most of the labs have BSL 1 and BSL 2, and it suffices most of our requirements, because we are not dealing with very dangerous viruses or very dangerous bacteria. So BSL 1 and 2 works fine for whoever is working on cell biology, developmental biology, or some kind of biomems. But those who are working with biosensors, those who are working with actually viruses, and they are working on different kinds of like infectious agents. So for them, biosafety level 3 and biosafety level 4 cabinets are actually very important. The biosafety level of biosafety or the BSL is actually for the hood that we are using, this kind of hood. So this kind of hoods are designed for different kinds of using for different kinds of microbes, or using different kinds of different cells. So depending upon what kind of cells you are working on, this food differs. Incubator is mostly the same for all kinds of cells, whether viruses or your bacteria, because when we are actually pelleting the cells, or when we are trypsinizing the cells, that is the time when the cells can actually can contaminate the surface. So the hood is actually the biosafety levels generally refers to what level of hood you are using. So if it is biosafety level 1 or biosafety level 2 hood, then it will look like something like this. Biosafety level 3 and 4 hoods are very, very advanced, and they so people need to wear PPE kits and gloves and masks and totally covered clothing, so that they do not get exposed to any kind of infectious agents that they're working with. So this is what a cell culture lab design generally looks like. So this cell culture lab design is actually it has here generally we have the inverted microscope. So whatever requirements mainly people have as the microscope, the cell counter, centrifuge for pelleting the cells. Some people will have water bath. So yes, deionized water supply. So here we can see that deionized water supply is there. So water is a very, very important source of contamination, because water that we generally use in our labs and everywhere, it is filtered through a lot of filtering machines. Though it is filtered through a lot of filtering machines, we actually don't know how much it is getting filtered or how much ions it is present. So ions present in the water can actually alter with your media composition. It can also alter with your pH of the media. So deionized water is generally preferred. So milky water. So there are different grades of water. We have distilled water, we have RO water, we have deionized water, we have milky water. So based on what are the advances of the, like what are the different kinds of like what is the basic requirement is to have contamination free deionized water. So people generally, many people, they will have deionized water inside the cell culture room. And so inside the cell culture room, when we are having this water system, that means we are not relying upon water from the outside the lab facility. So this actually helps in, you know, this actually helps in eliminating some chances of contamination. So this is required for, you know, like people will prefer that, but not all people can afford the water supply and deionized water supply inside the cell culture lab. Apart from that, we also need to have the water purifier, not water purifier, the cryo freezer, or the you know, the liquid nitrogen cylinders. Liquid nitrogen cylinders are required for storing the cells for a longer duration. And also apart from that, the autoclave that is present, sometimes we generally autoclave outside the cell culture room because we don't have so much of space inside the cell culture. Also the incubators and the carbon dioxide incubators, carbon dioxide cylinders, and all the hood and everything. So this is generally what we should have. And what people should generally avoid is keeping the hood just next to the entry. So this entry, just opposite to the entry, we should not keep any kind of hoods or incubators, because these are the sources of contaminating air, and it might enter the hood, or it might enter the incubator and cause you know, a problem. uh, so those are things. And also we should have some kind of refrigerator, and freezers, because there are a lot of chemicals that needs to be stored. And cell culture chemicals, it is better to store it inside the cell culture room, because it is not you know, accessible to always go outside the cell culture room and you know, take out the cell culture consumables and perform the cell culture. So always cell culture refrigerator and freezer needs to be separate from what we are using in the bench. Then these are some of the images that I took of what are the difference between the vertical airflow cell culture hood and horizontal airflow cell culture hood. So vertical airflow cell culture hood has the HEPA filter over here, as I already talked about. So this is the HEPA filter. It should be present in the vertical region. So it should be present in the top portion of the hood, and air will be flowing from the top like this. And this is the horizontal layer cell culture hood, where you can see that this HEPA filter is present in this region of your hood, and the cell and the air is passing in this way, like the airflow is towards the outside part. So what it is doing is, it is taking out air from the inside the hood and not allowing the outside air to come. So the pressure of the air from inside is so high that the outside air is not able to enter the hood. Also from here also, the air is coming out in such a, it's like a waterfall. So it comes out in very high pressure form. You can, like when you put your hand inside the incubator, you can feel the air coming out. So the outside air generally does not get chance to enter, but it is not avoidable that your air cannot be contained, right? Air will have some place to enter inside this kind of setup. So it is always necessary to sterilize the surface and use sterilized consumables before starting cell culture. This is the cell culture incubator. This is how it looks from the inside. So in cell culture incubator also we have HEPA filter over here. So this small uh, filter is known as the HEPA filter, and these are three different racks. So the incubators, they have racks depending upon what size of incubators you need. So these are three different racks you can place your cells, and here you can see a tray kept. So this tray has water inside it. So this water helps in maintaining the humidity inside the incubator, and it helps in maintaining that physiological humid 37-degree temperature and the humid bicarbonate buffer system inside the cells. Next, also we have what is called the liquid nitrogen storage containers. So this liquid nitrogen storage containers looks like this. So these are different sizes, big size, small size, whatever size we require. And liquid nitrogen storage containers, they in the inside they look like this. So inside this we have four columns. Inside big ones we have four racks, and in four racks, like this big racks, we can have small cryo boxes, and inside these boxes we have the cryo vials where we put our cells and freeze it. So liquid nitrogen, we have to continuously replenish it because it gets evaporated. So this liquid nitrogen is placed in this in the container, and it is generally, when we replace it, it has to be filled. It has to be till here, and like totally immersing the entire cells inside liquid nitrogen. But whenever we open the liquid nitrogen cap, you will see that the vapors are coming out because it gets evaporated very quickly. Also, apart from this, we require microscope which can actually detect, preferably detect all the bright field, phase contrast, DIC, and fluorescent pictures. Because if we want to see whether a transfection has happened, which has a fluorescent tag in it, we should be able to see it in the cell culture microscope itself. That okay, so transfection has happened. Now I can proceed with the experiment. Or whether we want to see what kind of morphology the cell has reached, then also we have to see it through the microscope. So these are the three different images I have taken of the same. I have not taken this, taken from the Leica Microsystems web page. So Leica is a company which makes microscopes. Leica, Zeiss, and Olympus. These are very good companies that make microscopes that are supplied worldwide, and we also have an Olympus microscope. So this microscope has the tendency, like has the features like it can detect cells in the DIC mode, in the phase contrast mode, and in the fluorescence mode. So the difference between DIC and phase contrast is DIC will give you a 3D like picture. See, you can see the cells a little bulged up. So it will give a 3D like image, and you can see what is the the thickness of the cells. This is generally required when we want to do something like 3D cell culture, or we want to see the cells in like how it looks in 3D, or how it or we want to use some electrodes to poke the cells. So if we are able to see the bulge kind of structure, it is very convenient to do those kinds of experiments. Otherwise, phase contrast is generally fine for checking what is the confluency of the cell, or whether the cell has grown or not, those. Also, apart from that, so for fluorescent imaging, also we want to see whether the cells have been properly transfected, or whether the cell have taken up the dye. So just quickly, to see whether cell has taken up the dye or the cell has been transfected, these things are required. So microscope inside a cell culture room is a very important requirement. Next is animal handling and ethics clearance. So the people who are actually working with animals, and they are working with primary cell culture. So I had discussed about what is primary cell culture in the earlier class. So again, I'll just go tell it. So whenever you want to dissect a tissue or cells directly from the animals inside the cell culture facility, that is known as, the first set of cells that we isolate from the animals is the primary cells. So those that requires a lot of sterilization apart from the normal cells, secondary cell line that we are culturing, because we are actually isolating cells from animals body, and animals, they are naturally prone to have a lot of microbes in their system. So animal handling, proper dissection procedures, and carrying out proper ethics is actually very important. And we need to maintain, and we need to follow all those rules and regulations very strictly, so as to avoid any kind of inconveniences. This kind of facilities are generally for a very common group of people. Animal house, or the dissection facility, or the animal handling rooms, and all these are for a group of people. So any kind of misuse or any kind of carelessness in that facility can lead to the inconvenience of not only just your work, but it will lead to the inconvenience to other members who are also working and using the same facility. So whenever we are using such kind of common instruments or common equipments, we have to be really careful to not mess it up and always follow the rules and regulations carefully. Whenever we are also, the safety regulations include the proper disposal of those animal waste, because we don't know actually what kind of treatment that animal was going through, what kind of drugs that animal was fed. So it is always essential that whenever we are working with animals, it has to be very properly disposed, it has to be properly like labeled, and not just thrown around like that. And it has to be properly sterilized. Ethanol spraying and UV sterilization is a must. Also, proper rules and regulations need to be followed. And the cell culture room and design layout needs to be very convenient and spacious according to the requirement of whatever animals we are working with. So generally, the primary cell culture facilities, they do not coincide with the secondary cell lines procedure, because they need to be two separate labs. One lab where people are culturing primary cells directly from the animals, and another lab where they are continuously growing the secondary cell lines. So as to not contaminate each other's environment. So this kind of separation is important, because primary cells, there is a lot of chances that contamination can happen there. There is so much of contaminating substances in when we are isolating neurons from the mice or any kind of animals. So what kind of sterilization do we need to follow? There are different kinds of sterilization: autoclave sterilization, UV sterilization, and filtering the apparatus, filtering the media. So most commonly used sterilization is all these three techniques. So in autoclave sterilization, we have to so all the tip boxes, all the tubes, all the tip boxes, tubes, the water that we are using for preparing media, or the water that we are using inside the cell culture incubators, inside the cell culture incubator, the water that we are using, everything has to be autoclaved. Everything has to be autoclaved, and it has to be properly dried in hot air oven. So there is so after autoclaving we get some. So how does autoclave sterilize? So autoclave sterilize using steam sterilization, and this steam sterilization means that autoclave water, whatever is present inside the autoclave, that also gets, like, droplets are there inside the tip boxes or inside the tubes and whatever we are using. So we need to properly dry it before using. So those things need to be maintained very carefully. So autoclave sterilization and UV sterilization is a must, along with the alcohol spraying. Also, I have not mentioned here, but yeah, alcohol spraying. So this kind of sterilization will help in reducing the amount of contamination that we generally, try to avoid. And so yeah, so this autoclave sterilization and UV sterilization are very essential and very important. So this is the cell culture hood. As you can see, inside the cell culture hood, there is pipettes, there are these pipette holders, and there are some tubes and everything. So we also generally prefer to keep our gloves and tip boxes also inside it, so that we can use it so before keeping it for UV, so that we can sterilize the entire surface with UV. So UV generally means surface sterilization. It's not going to penetrate deep. It does not penetrate plastic. It does not penetrate any substance. It is just for surface sterilization. So for pipettes and for tips and tubes, and so we open the box of tips and we open the gloves which we want to use. We put it in the hood, and then properly wipe it with ethanol. Everything we have to wipe it with ethanol, and then switch on the UV. After switching on the UV, we wait for 20 to 30 minutes. Apart from the hood UV, there is also room UV that labs are generally they have. So room UV and hood UV both needs to be switched on. before we proceed with our cell culture. So yeah, so this sterilization is surface sterilization, and another sterilization technique that we use is filtering. So any kind of chemicals that we use inside the cell culture, whether it is media, whether it is water, whether it is PBS is a buffer, whether it is any kind of like treatment you want to give, FBS, anything, whatever goes inside cell culture needs to be filtered. This kind of chemicals, many chemicals cannot be autoclaved. Like for example, FBS. For example, FBS is fetal bovine serum. So fetal bovine serum is used for as a like serum it has all these amino acids and proteins as a serum component of your like blood. So it provides all those essential nutrients to the cells, but this kind of FBS cannot be autoclaved, because if you autoclave it, all the so FBS is a serum containing serum containing all the proteins. All these proteins will get aggregated, and those aggregation we don't want. We generally know that at higher temperatures proteins can get aggregated, so we don't want it. So we cannot sterilize it using the autoclave. So then we use filtering apparatus to filter it. So in this filtering apparatus, you can see there are two components. One is the filtering bottle in which the filtered media is going through, and the other is the bottle which has the unfiltered media. So this kind of apparatus is actually attached with a pump. So this pipe you see, this pipe has a pump attached to it. That pump will pump the air and help in, this pump will help in passing the liquid through this filter at a very quick amount of time. So if we wait for gravity for the filtering to take place, or if we put some pressure, it will take a lot of time. This is a lot of media, around 500 to 1 liter, 500 ml to 1 liter of media needs to be filtered. So this kind of pumps are very essential for filtering the media, so that any kind of particles that are remaining inside the media has to get removed, and we do not want any contamination in that source. Media is generally needs to be prepared under supervision, because we do not want the main media to get contaminated, or else whole cells, whatever you have in your incubator, will get contaminated. So this filtering apparatus needs to be, but the filtering apparatus that we are using here, this needs to be autoclaved, and this needs to be sterilized before passing the media containing media and FBS through these filters. So yes, that is now for today's lecture. Any kind of questions you have before we move on to the practice assignments?
Ma'am, how do we sterilize the CO2 incubator?
CO2 incubator, we can fumigate. So CO2 incubator, what we do is inside the CO2 incubator, there is a CO2. So inside the CO2 incubator, we can wipe the entire surface. So we have to first switch off the incubator. If it is working, we cannot fumigate it. So we have to first wipe the incubator with ethanol. But this glass should not be wiped with ethanol, because this glass is sensitive to ethanol. So we cannot wipe this glass with ethanol. So entire surface inside the incubator we can wipe with ethanol, and then we have to put methanol. So methanol we have to put in a proper 60 mm dish or a 35 mm dish which is sterilized, like from sterilized packet. We have to put it inside the incubator, close the door, and keep it for 1 day. Methanol will get evaporated, and this methanol acts as a disinfectant, and it will smell very bad. So after 1 day or 2 days, we have to take out the container where we kept methanol, because it will be evaporated already. And then we have to wait till the time that smell gets disappeared before we start with cell culture again. So methanol fumigation is required for incubator, sterilization. But this is not done on a very frequent basis, because fumigation is also not a very healthy, means it's not very good, good for your health. It is very toxic. We use methanol, we use potassium permanganate, formaldehyde, and everything. Those are really very toxic chemicals, and we should not use it. We should not come in contact with it frequently. But yes, if after every 2 to 3 months, we need to fumigate it.
Yes, ma'am.
Okay. Any other questions?
Which
Ma'am,
kind of laminar hood is better for the cell culture, vertical or horizontal?
So it is vertical hood is I think better, because vertical hood your airflow is not like coming towards outside. It is like flowing like a waterfall. So it is not very inconvenient, in the sense it is not like the air will not be in your, like when you open the hood, it will not come to your face like directly. And also this kind of hoods that are like what horizontal hoods, these are generally used for bacterial cell culture, because this is better for like bacterial work, bacterial part. But and also this part, if the HEPA filter is in this part, you cannot actually keep anything here. Like basically many times we try to keep the tip boxes, or we try to keep, there is a lot of things that we need to keep inside the hood for working, the tubes or the stand or pipette holder. We cannot keep anything here because it will be blocking the way of the air. So everything has to be kept in this portion of the hood. But in case of vertical, you can use the entire portion of the hood. Like you can keep the chemicals or keep the tubes everywhere here, and you have a lot of space to work with. So ideally, and it should, like, better is to use the vertical one. But vertical one, the problem is the vertical's height is very high, and so many times it does not fit the cell culture room. It is not very portable also. It is like once you have set it in one place, it has to be there only. But this kind of systems, these are very portable, and they have wheels in below the stands. They have wheels, so you can easily change the position if you are not comfortable, or if there is some problem. So for cell culture, generally we would like to prefer the vertical ones. We also have the vertical ones only in our lab. So
Okay.
Ma'am, can we install UV lights inside a CO2 incubator for sterilization?
no, actually UV light, if you want to install it, then we need all the switches and all the connection. So inside the incubator, we don't have any kind of electrical, inside the incubator, we don't have any electrical connections to, externally install UV light or something. So, and also incubators, no, they, if you are using UV light, so it will ionize it. Like UV light inside the incubator, any kind of radiation is not actually very healthy for incubators, because it will be altering with all this temperature, all the water, like water and the media that we are using in the cells medium. So UV inside the incubator is not a very good choice, because like ionizing radiation is very harmful. I mean, in inside the incubator, it will alter a lot of properties, I guess.
The water what we use for cell culture, is it required to be millipore water or
Hmm.
deionized water is sufficient?
It has to be milky water. For cell culture, always milky water is required.
Okay.
Everything free, like totally very highly purified and autoclaved as well.
After milky water also supposed to be
Yes.
autoclave?
Yes.
Okay.
After milky water is autoclaved.
Okay. Thank you.
So any of you have done cell culture, like practically?
I'm working with bacterial cultures, so these are a little bit similar to me.
Okay. Okay. Yeah, bacterial culture also we need to have certain precautions, but in cell culture you have to have even, I mean, a really high level of precautions. It is not like very kind of tricky, but we have to be very cautious with everything, whatever we are doing, because we generally don't try to keep in mind those things when we are working in like lab for other purposes. But whenever we are working with bacteria cell culture, we have to be really cautious that okay, not to touch anything outside, not to put our hands in like places without like once we have put our hands outside the hood, we have to always spray alcohol and then only put our hands inside the hood. So those things, like small things, they are very much important for cell cultures. Mm. Darshan is asking, so is DIC mode of microscope similar to Z-stack microscopy? So Z-stack is basically Z-stack is not a type of microscopy. Z-stack is a property of all the, you can use Z-stack for DIC, you can use Z-stack for fluorescence, you can use Z-stack for bright field, everything. In DIC, what happens is your your image itself will appear bulged because there are some differential interference, like some contrasting they're doing with the background and the cells, as a result of which your background is reduced to such a less level that your cells appear to be bulged naturally without doing Z stacking. Z stacking, what does it mean is in that cell, when you're doing the imaging from this top layer, like suppose this is your cell, the entire cell, and the imaging is from each of these layers. So each of these layers, then what will happen is 3D image you will get. You will get a 3D image of the entire cell. That is not what is similar to DIC. DIC is a type of microscopy. Z-stack can be used in DIC also. It can be used in fluorescence also. It can be like layers of imaging. What we do is we see the cells from the top. Like suppose the cell is like this, in like rounded cell, we see from the top, and if it is bulged, we will see from the top as bulged. In Z-stack, all the stacks will be like all the layers will be stacked in thin layers, so we get a 3D image that way. Yes, Koshi, I will upload it. Actually, I have not uploaded the last week's recorded version also. I'm really sorry. Actually, I was caught up with some work, so I did not get the time. I will upload the recorded videos and the also the slides also together. Maybe I am trying to upload this week only. So yeah, don't worry about that. So let us go to the questions. So this is a question from uh, previous class when we were doing our cell cycle phase. So which of the following is not a phase of cell cycle? You can answer in the comment box. That's right. N phase is not a phase of cell cycle. We do not have anything called N phase. So we only have G1 phase, G2 phase, S phase, and M phase for the mitotic phase. Let's go to the next question. Yes, everybody has given correct answer. So very good. Option C. Yes. So it's correct. The which of the following stages of the cell cycle the cell copies its DNA? It's the S phase or the synthetic phase. So this is the correct option. So in G2 phase, G2 phase and G1 phase, cells actually help in cells in all the accessory parts, proteins and the lipids and all other material, or all other material except the DNA. Everything is doubled in G1 and G2 phase. Only the DNA gets doubled in the S phase. Option A is correct. So in cell culture, the extracellular matrix coating on a substrate is required for what kind of cells? That's right. So in cell culture, the extracellular matrix coating on a substrate is required for the adherent cells because the adherent cells are generally, it will be growing on this lower portion of the dishes. It will be attached to it, and this portion has to mimic the extracellular matrix that is present inside the cells. Non-adherent cells, they are not, getting attached to any kind of substrate, so it should not like cause an issue whether the extracellular matrix extracellular matrix coating is there or not. We are not concerned with that. We generally want like the entire media only is it will grow in suspension kind of thing. It's not going to get attached. Immune cells, the WBC macrophages and all those cells are generally non-adherent cells, so they are not going to be requiring the extracellular matrix coating. So the correct answer is adherent cell. So next question is, which of the following is usually a marker of pH in cell culture conditions? Correct. So everybody has given the correct answer only. The usual marker of pH in cell culture condition is color of the medium. So here you can see that the media is red color, right? So this red color comes from phenol red. So phenol red is an indicator Phenol red is an indicator of what is the pH of the media. So if it is basic, it is going to be red color. If it is acidic, it is going to get yellow color. So generally in our media, we have sodium bicarbonate. So in sodium bicarbonate is a basic compound. So it will give red color to that media, but this gets depleted with, if cells are growing and using that sodium bicarbonate, it gets depleted and form carbon dioxide. So then it will, if it is carbon dioxide is released, then it's going to turn acidic, and that is going to be yellow in color. So the red media will turn yellow, and that is usually the marker of pH, that the pH has gone down, and we need to change the media, and we have to, replenish with new media. So moisture levels, no, moisture levels is not a indicator. Cell size also no. Osmolarity is also no. Correct answer is option B. So Karthi is asking, the cells are cultured in a media with phenol red. If the cells are metabolically very active, the media will turn yellow. Is that correct? Yes. So if it is very active, in the sense if it is growing very fast, I mean some cells they grow very fast. Within one day, it will be totally confluent. So if those kind of cells are there, in one day your media, red color media, will turn yellow. But generally, what people do is, for their convenience, they will try to seed a little less amount of those cells, so that at least one day of rest you can get before next day. Otherwise, every day you have to come to culture your cells, and you have to change the media. That is also not very economical, right? If you have to change the media every day, that means you have to make a lot of media all the time. And making media is also like really very, you have to be very cautious while making the media. So metabolically active cells, they turn quickly red, from red to yellow. But yes, we have to optimize in such a way so that it does not grow at a very fast space. I mean, we can get some time between uh, two sets of cell cultures. So yes, now next question is, so which of the following parameter will change if carbon dioxide levels go low in an incubator? Yes, Samad, actually, I did not get the time to, upload the recordings and PPT. I've been a little busy last week. I will definitely upload it as soon as possible. I'm trying to upload it by this week, so I will be uploading it. I'm really sorry. Yes, Samad, definitely will get the recordings. Don't worry. Okay, so this question answers from Gangotri. It's not temperature. If your carbon dioxide levels go low, temperature in the incubator is not affected. Okay, temperature is definitely a different. It's a different parameter altogether. It's not going to affect just because carbon dioxide levels go low. Because in carbon dioxide, what do we generally know about carbon dioxide is, in carbon dioxide is a acidic substance, right? So it is like carbonic acid, we say, or carbon bicarbonate is a acidic. It will go to acidic or basic levels. Like it's going to be altering the pH only. going to be, or concentration of the growth factors, nothing. Answer C. Like the option C is not correct, because concentration is not going to be, concentration of the growth factors is not going to be affected by carbon dioxide levels, because carbon dioxide is only going to affect the pH. So the option correct is option number D. So Karthik is asking, carbon dioxide incubators are used for cell culture for maintaining humidity or pH balance. So carbon dioxide incubators are used for maintaining temperature. It is used for maintaining humidity also. It is also maintaining the pH. So three things only it is required. It is maintaining carbonate buffer system. Okay, carbon dioxide incubator is used for maintaining the, so in our body we have bicarbonate buffer system. So it helps in maintaining that, and it also helps in maintaining a temperature at 37 degree. Also, it helps in maintaining the humidity, I mean the humid environment. So three things only. Okay, so next question. Okay, so here a lot of people, they are answering all of the above. This is not correct. See, I will tell you. So if we are switching on UV bulbs while working inside this incubator, while working inside the hood, it is not good at all. UV light is not at all good for your skin, or not at all good for your health. So during working, UV bulb should be switched off. So before working, UV has to be on, and like it has to be sterilized for 20 to 30 minutes. After your sterilization with UV, 20 to 30 minutes, when you go inside the cell culture room, UV has to be off, and then only you have to start working. So this option is not correct at all, that UV bulb has to be on while working, because it is very like hazardous. This option, turning off the blower while working inside the biosafety cabinet, this is also not correct, because blower, what it does is it helps in that air flow. So here that I had discussed about how the airflow should be in the kind of hoods. So if you're when you're switching off the blower, either the airflow is vertical or either the airflow is horizontal. So if you're switching on the blower, the airflow will start taking, like airflow will be affected. So if you are switching off it, if you're switching it off, then that airflow will not be maintained. So your the air that has to be taken out from the incubator, from the hood, it will not be maintained, and as a result of which the air from outside will enter the hood, and that will cause contamination issues. So this option is not correct. Turning on the blower is correct. So this is not off, this has to be on. This has to be on. So only option number A is left. So option A is only correct, that we have to spray the items with 70% ethanol always. Whenever you're doing cell culture, it is very, very mandatory that you have to make ethanol your best friend, and you have to always wipe your hands with ethanol. Always, whenever you're taking out your hands outside the hood, wipe your hands with ethanol and then only put it inside. Or if you're touching the incubator, touching the microscope, touching anything, practically anything, you have to use ethanol. Wipe all the surface with ethanol before using the cell culture facilities, and then you have to continue with your work. So option A is correct. Next question is, Yes. So everybody has given the correct answer. So option number B, correct. So aseptic techniques we are required to prevent contamination in cell culture. Okay. So be careful with what words we are using. Okay, because there might be some confusion with ensure contamination, prevent contamination, ensure non-sterility. So yeah, be careful. So option number B is correct. Next question. Hmm. So here also there is some confusion. Lot of people have written B, B, B. So yeah, let me. So what are basically pyramidal neurons? Pyramidal neurons, as the name suggests, it's a neuron. So it's going to be like these are present mainly in the cerebral cortex, in the brain region. So this is not going to be correct. That is right. Microglia, these are glial cells present in the brain, which helps in providing support, nutrition, and etc. So these are also not going to be present in the. So these are also going to be present in the brain. So that is, not the correct option. What is astrocytes? So astrocytes is actually a type of glial cells only. Okay, this is a type of glial cells which help in these are not associated with neuronal transport of neuronal impulses, but they help in the accessory functions. They help in clearing out of excess neurotransmitters. They help in providing like nutrition. They help in promoting the synapse growth, synaptic connections. So these are also present in the brain only. Astrocytes is also a like brain cell type as well. Only Scavens cells that is there. These are not present in brain. These are accessory neurons. These are like accessory part of the neurons, generally present in our spinal, like your not in the central nervous system, but in the peripheral nervous system. The Scavens cells are present, which help in providing nutrition and help in providing like support to the cells. So Scavens cells is the correct answer and not astrocyte. Okay, this is not the correct answer. So option number D is correct. So next question. Hydrophilic surfaces for using. So yes, most of you have given the correct answer. So option number B is correct. Cells prefer to generally attach on hydrophilic surfaces because hydrophilic surfaces they have the water like some patterns they follow, as a result of which it helps in promoting adhesion. So uh, like we generally know that if you have water, water has the property of cohesion and adhesion. So if the surfaces are hydrophilic or water loving surfaces, it's going to promote adhesion and it's going to promote the cell surface attachment. If it is hydrophobic surfaces, it's going to have a lot of repulsion to water, so it's not going to support uh, the cell attachment. So the correct answer is option number B. So, yes. So that is all for questions. Any other doubts? Please let me know. Anybody has any other questions? Okay. So it's not then we will say bye. Also, I think yeah. Okay. Okay. Bye everyone. Oh, Dhanalakshmi is asking a question. What are the major factors involved in the contamination of cell culture? So major factors, it's generally uh, practicing like if we are very careless, in the sense that if we are not washing our hands properly, if we are having cold and coughs and still we're entering the cell culture facility and coughing, if we are not using alcohol frequently, those are the major factors which are involved in contamination. Some of the factors also include that if the cells, if the cells are from the stock only, it is contaminated, like the cells that you have received from somebody else lab, from somewhere else, those are contaminated, so it will lead to contamination of all other cells. So those are the some important like major contributing factors, but generally it is due to lack of caution and precautions that contamination happens. So Giotama is asked to, is there any other buffer system except the carbon dioxide buffer system in the cell culture? No, there is only carbon dioxide buffer system, because in our body we have bicarbonate system, buffer system. So always to mimic the bicarbonate buffer system, we have the carbon dioxide buffer system in cell culture, because we want to mimic as the physiological, it has to mimic the physiological conditions inside the body. That is the reason. Samat is asking, out of this, which factor should be included while choosing a hood for cell culture facility? Noisy, high vibrational levels, no ultraviolet light facility, at least per space per person. I think for cell culture facility, the most important criteria is how better it is able to, how better is the BSL level. I mean, if the BSL level food is better in the sense that BSL2 or BSL3, then the airflow or air circulation is the best, and this air that is going to enter from the outside will be very difficult to enter inside the hood, and that is going to protect from a lot of contamination in the future. Also, no ultraviolet light facility is definitely a big no. You always need a ultraviolet light inside the hood. And noisy vibrational levels is not a much issue. At least 4 feet by 2 feet working space per person. I generally like hoods which have like personal spaces, because you are not altering the work of other person. If two people are working in the same hood, and if any one of person loses his or her caution or precautions, then the work of both the people will get affected. So it is always better that only one person at a time is using the hood. So a smaller hood but with better facility, having UV, having proper BSL levels, those are, I think, the best requirements. For how long do we have to incubate the cells inside the carbon dioxide incubator? It depends upon what is the growth time of your cells. Some cells grow in 24 hours. So some cells duplicate in 24 hours. What is the duplication time? So it depends upon how fast your cells are growing. You can keep it in your carbon dioxide incubator to allow the growth. If it is slow growing, you can keep it 2-3 days, no problem. If it is fast growing, at least every day once, or every 1.5 day, you have to check what is the status of your cells. So yeah, depending upon your cell type and cell growth, it depends. Does the carbon dioxide interact with the cell culture? Yes, definitely. Carbon dioxide in the incubator does not interact with the media directly, but carbon dioxide along with the water that is present in the incubator help in maintaining the carbon dioxide buffer system. Like the H plus and the like water has H2O, no? So the H2O will be H plus and OH minus. So those H plus will again go with carbon dioxide to make the carbonic acid, and those carbonic acid will be in the inside cell culture incubator, and those carbonic acid or the carbonate system will help in maintaining the pH of the maintaining the bicarbonate buffer system inside the body, inside the cells. So Diotima is asking, when we autoclave, how does the lines appear on the autoclave tape? What is the principle appear? How does the lines appear in the autoclave tape? We don't have autoclave tape. We have an autoclave bag. In the autoclave bag, we will put all the tip boxes and we will close it with rubber band. And so the lines that is happening is due to the rubber band that is getting, when we have to tightly close the, bag, so that water does not enter the bag, autoclave bag. So only steam that is present is like steam should be there, but no water or additional things should not enter the bag. So yeah, so there are no lines as such, but yes, it will get creased because you are tightly binding, tightly like closing it with rubber bands. So those things. We use autoclave tape in your lab, so wanted to know about it. Okay. In our lab, we don't use autoclave tape. We use normal rubber bands. The rubber bands and close it. So that is okay. Okay, we don't use tape actually. We use rubber bands. That works fine, I think. How do the cells get the oxygen in the carbon dioxide incubator? Oxygen, how does it get the oxygen? This I have to see. I am not very sure. How does it get the oxygen? I think from the water only. I mean, the water that is present, it will have H plus and OH minus. Those OH will get converted to oxygen system. I am not sure. I'm not very sure about it. Do cells? I have to check. I'm really sorry. On the material to be autoclaved due to humidity, magnesium is an acellular component in human body. Yes, ECM is an acellular component in the human body, correct. So it is basically a matrix. It is a matrix of proteins, lipids, carbohydrates, everything which is present outside the cells and provides nutrition for the cells to grow. Yes. What is suitable buffering system for cell culture? What provides a suitable buffering system for cell culture, Isha? You're welcome, Preeti, Sakshi. Okay, any other questions? Yeah, so which factors should be included while choosing a hood for cell culture facility? Noisy, high vibrational levels. Those are noisy or high vibrational levels, no ultraviolet light facility at least. Uh, so these are the options you're saying. Noisy, high vibrational levels. I think the fourth option, 4 feet to 2 feet space per working person. Last option. Yeah. No, we don't. If you have an option for not choosing a noisy cell culture hood, then you should not choose a noisy cell culture hood. No, I think option D is correct, Samar. Yeah, option D. Okay. So if there are no more questions, then any more questions, anybody? Okay, everyone. Then we'll meet again next week. Thank you, everyone. Again, we'll see you. Bye.